everybody. I hope you can hear me. My name is Professor Judith Breuer. I'm from University College London. I'm Professor of Virology, and I'm also a consultant in virology at Great Ormond Street Hospital for children. And welcome to this webinar on EBV disorders with EBV involvement. And we have three very interesting speakers today. The first one will be Cristina Venturini from UCL. Cristina is a computational geneticist at UCL. She specializes in viral genomics and she has expertise in chronic viral infections, which she's interested in, which has led to her interest in EBV. And she is particularly interested in the evolution of viral infections in under chronic conditions. So thank you very much, Cristina, and I will turn my camera off, and please feel free to start when you're ready.
Great, thank you, Judy.
Actually, before you start, Cristina, can I just remind everybody, if you've got questions and questions, please can you put them in the Q&A? Don't put them in the chat. Please put them in the Q&A. Sorry to interrupt you, Cristina. Please carry on.
That's okay. Hopefully you can see my screen now, my presentation. And so today I want to present some of the results about how we look at the viral genomics to look at biomarkers that can help us understand the management and understanding of EBV disorder. So yeah, okay, perfect. So I think now you can see my screen now. And I'm just going to put the laser pointer on now. Perfect. So I work in a group with Judith Breuer in UCL and in the Department of Infection and Immunity. Because I'm the first speaker of the day, I just thought I remind you a little bit about the EBV infection cycle. So EBV is transmitted by saliva. And the main target cells are the B cells that express the CD21, and the virus can also infect the epithelial cells and can initially replicate the oropharynx before the infection in the B cells. Naive B cells, they become infected in the mucosal in the mucosal lymphoid tissue. And they can then establish a pool of latency and infect the in-memory B cells. What happened in the EBV is that after the primary infection, we can have a periodic reactivation of latency infected B cells, and then they sort of reactivate again in the saliva. And the reactivation in the saliva, the replication in the saliva is absolutely necessary for transmission. Due to the low expression of the viral protein, infected B cells escape the sort of like immune control. So in these slides, I just wanted to kind of give you a little bit of overview of conditions that are associated with EBV viremia and or different EBV tissue. So we have quite a big continuum of diseases. We start from a sort of asymptomatic condition, which again, in the immunocompetent host that can be asymptomatic or also can show as an infection mononucleosis. And then we have situation when we have an immune dysregulation, and this can be, for example, it can sort of lead to situation like chronic active EBV or sort of other type of NK and T cells lymphoma. But also we have other type of malignancy like the B cells lymphoma, Hodgkin lymphomas, and also we do have other type of malignancy that can also happen in immunocompetent host. This is just like a slide to remind you that EBV can affect both can affect the immunocompetent, but also can affect. I mean, also like it can be like mostly affects people that have some immunodeficiency. So for this study, we've been working mostly with Great Ormond Street Hospital, which is a pediatric hospital in London. I'm actually going to mention that I'm sort of presenting two part of a study. So the first part, we've been published that in 2021, and then we have a slightly newer part of the research project that is still in progress. What we wanted to do was to look at EBV across different conditions. So we have some sort of like the healthy, which we have some infection mononucleosis and also an asymptomatic shedding. And then we have different conditions that goes from post-solid organ transplant. And also we have some primary immunodeficiency, some chronic active EBV, HLH, and other type of lymphoma. And just to remind you that for the chronic active EBV, we refer to the definition that was in 2023. For the first part of the study that we published in 2021, we mostly analyze sample from saliva, from infection mononucleosis, and a few other conditions. And these all affect different cells. So of course, we have involvement of epithelial cells, of B cells, of NK and T cells. And the first thing that we did was to look at, so of course we did some deep sequencing, and we wanted to understand more about the viral, what's the virus, so what we can understand from the viral genome. So when we look at, the first thing we look was at possibility of mixed infection. This is when we have an individual that is affected, that is infected with more than one strain or EBV. And this is something that we can definitely see with sequencing. We then look at the single nucleotide variants and also deletions. So when we focus on mixed infection, we actually see these are quite rare in EBV infection, but they are actually seen most likely in the saliva, or especially in the saliva samples. And this happens in both the healthy, but also in the chronic active EBV saliva samples. When we look at the single nucleotide variants, these are present in various conditions and in both healthy and in non-healthy, in the saliva, but also in the blood. But they are not, they're seen a lot of fewer of these type of variants in the PTLD and in the solid organ transplant, probably due to the fact that EBV is clonal and not replicating in those cases. We then look at the deletions and we split the deletion in either smaller deletions in the EBV or EBV deletions that are quite big, so these are over 2 kb. And what we see is that the smaller deletions are quite common in all samples, both in the healthy and in the non-healthy. But when we look at the bigger deletions, these are only present in samples from patients with immunodeficiency and chronic active EBV, and also in tumor samples. And these are mostly found in two specific areas of the genome, and these are the C promoter and the BART microRNA. So just to give you a little bit more details about this study, when we look at the small deletions, and here you can see on the y-axis, on the y-axis, you can see the different parts, the different genes and the different part of the BART microRNA. On the x-axis, we can look at the healthy and the mononucleosis cases. And we can see, for example, that we can see a lot of these small deletions happening, especially in the latent genes. And then when we look instead at the more like at the BART microRNA, we can see that those are mostly involved in the malignancy, in the tumor sample and in the Hodgkin lymphoma. When we look at the larger deletions, we can see that these are not as common in the healthy individuals. There are still some that are mostly happening in some of the latent genes and in other parts of or elite of the genome, but they're not as common. And we can then see that in the chronic active EBV in the tumor, we can see that we can see deletions that include the BART microRNA and also the one before in the latent genes. And especially we can see in the tumor samples that's a lot more deletions compared to the other samples. So what I just wanted to point out here, when we did these studies, more or less at a similar time, there were a few other groups, mostly from the group in Japan from Murata, and this was a Chinese group that conducted similar studies looking at different EBV-related diseases, like chronic active EBV and other type of lymphoma. And what they found is that they show exactly similar deletion compared to ours. So they found that especially 35% of chronic active EBV had deletions in this area of the genome. And they found similar percentages that go from 35 to even 71% of samples with similar deletions. And this Chinese group found similar results also for the NK T cells lymphoma. We then wanted, because this was very fascinating, and we wanted to look at additional samples, and also we wanted to take advantage of the fact that we work closely with Grey Ormond Street Hospital, and we can have not only samples from these patients, I mean, we can also have longitudinal samples, and we can have information and metadata about the treatment. So when we went back and to look for additional data and to expand our patient cohorts, we found 2 more additional cases of chronic active EBV following again the 2023 definition. And we have, so in different color, we can see the different patients, and we have different, several longitudinal samples in the y-axis, and the samples also come from different compartments. So we have the whole blood, but we also have this different fraction. So we have, for example, the T cells, the NK cells, and we could sequence all of them separately. So what we found is that when we look at the C promoter deletions, this was found in both cases. We also found that one of these cases had the BART microRNA deletion, and we also find a lot of other deletions in the rest of the genome. When we look at then the primary immunodeficiency, we managed to collect 5 patients. And again, this from different compartments, from blood, T cells, B cells, and saliva. We found that also the primary immunodeficiency samples had some deletions. And these deletions were found again in the C promoter. So we have the 3 out of 5 had the C promoter deletion, but none of them, very interesting, didn't have the BART microRNA deletion. But they also were identified like small other deletions in other parts of the genome. We also had a few cases with HLH, and these were 6 in total. And again, we found that half of them had the C promoter deletion here in red. We then found that 2 of these patients had the BART microRNA, and then all of them had some other type of deletion. I wanted to point out also that in this patient in yellow, where we found the BART microRNA deletion, we can also see that over time, the patient started with a specific deletion that were consistent among 3 samples. The deletion then in a certain amount of time disappeared, and this is overlap with some treatment at the time. But then the deletion came back, and it came back as a different deletion, and in this case was actually a much bigger deletion, still overlapping with the same genes in the same BART microRNA region. So this was very interesting to see, and it was present in the blood cells, in the T cells, and it was very interesting that it was consistent, but it appeared in a different. It was a different deletion. We then also had a few malignancy cases, and these were all T or NK tumors, and 3 out of 4 had like a C promoter deletion, and one of them also had the one the BART microRNA deletion. And this is the same patient I showed you before. So this was a patient with HLH and also with that then developed malignancy. So just to summarize a little bit these results and look at what happened longitudinally, we have that most of these conditions that where we have a high persistent EBV show deletion in the C promoter and or in the BART microRNA, and also show deletions in other genes that, however, we don't really know what they mean and what it means. All of the patients, of course, receive some course of treatment, which could be transplant, rituximab. And then when we look at what happened next, we actually see that 7 of the patients where the deletions actually ended up disappearing, actually the outcome, they did well. It's true that we couldn't follow up, we lost follow-up 2 of these patients, but overall, all the 7 patients did quite well. The 2 patients where the deletion actually stayed there for a longer period ended up, one unfortunately died and the other had still recurrent EBV colitis. So this can give us a little bit a sort of idea that perhaps we could use these deletions as a biomarker to see how the treatment is working. So very recently this year, we also found there was this paper published that sort of confirmed the results of our smaller study. This was from the Japanese group that did the initial study as well. And they put together 990 EBV genomes from healthy volunteers, other EBV conditions. And they found that quite a few of the frequency of the larger deletions in a condition like chronic active EBV or other type of lymphoma was higher compared to the infection mononucleosis or other type of epithelial malignancy, or also compared to PTLD. And they also try to give a little bit, like trying to understand a little bit better what might be the significance of these deletions. So for the BART microRNA deletions, we think that this can promote the elytic phenotype, and this will create a faster cell growth and oncogenesis. And there are some mouse models that show that deleting the BART microRNA will give like a sort of like bigger tumors. They also show that C promoter deletions, but also not just deletions, also inversions are quite common. And this actually knocks out the EBNA 1 and 2 and promotes an immune evasion. They also show that the EBNA 3B deletions can reduce the tumor suppression functions, and therefore they promote oncogenesis. So the question, of course, is how this link to the pathogenesis. For now, our data fit and the data from the Japanese group link well with the biology. Chronic active EBV, HLH, other EBV malignancy are associated with expansion of myeloid-derived suppressor cells that are not due directly to the infection by EBV. These cells reduce the function of CD8 plus T cells. What we think might be happening is that the clonal expansion of EBV positive T and NK cells with these particular deletions might evade the suppression, particularly when we have this reduced CD8 plus function. Just to remind that EBV is present as sort of different populations, so we still have a replicating virus in the chronic active EBV blood and saliva, but we also have the defective virus in the T and NK clones in the blood. So what's also other of the cell of origin studies and the presence of different blood cell phenotypes is showing that all of these support the notion that EBV may in fact a common lymphoprogenitor, but not all cells are affected. Of course, these are at the moment not all of these, we will need further studies to confirm that. So this is all for me for today. And I would like to acknowledge quite a lot of people, but I keep it quite short. So a lot of this work has been doing in Judith Breuer lab. And in particular, I would like to thank Joanna, who was one of my master students that did quite a lot of the work. And also, all of this was possible with the help of people in Grey Ormond Street Hospital, and in particular Zainab, that helped us to put together all the metadata. And I would like to thank also the pathogen genomic streams of UCL Genomics, where we did all the sequencing, and of course, all patients and families for the help with this type of projects. Thank you.
Thank you very much, Cristina. Can I remind everybody to put any questions that you have in the Q&A and not in the chat? So now we'll move on to our second speaker, who is Sylvain Latour. Sylvain is works on the immunology, cell biology, and the immune system of immunodeficiency. He is particularly focused on natural killer cells and lymphoproliferative disorders, and his studies deal with areas such as NKG2D, X-linked lymphoproliferative disease, and the lymphocytes, as well as the in the immune system. He's very well known in the field, and his talk today is going to be Pathways of EBV Immunity Revealed by IEIs. Thank you very much. If you'd like to turn your camera on and start your talk.
Let's turn on, I think. You see me?
Yes, we can see you, and your slides are fine. Thank you.
Okay, so I can start. So thank you for the invitation. So I'm happy to discuss with you EBV immunity through the inborn error of immunity discovery. So my lab is mainly working on primary immunodeficiency associated with a high susceptibility to EBV infection and viral disease. And as you will see, this primary immunodeficiency represents a very unique model to understand the molecular requirement of T-cell proliferation and expansion during the immune response. So I will skip some generality about EBV because we have a nice presentation by Cristina. So regarding the immune response to EBV infection, so EBV, the primary infection occur in the tonsil, and the target cells are the epithelial cells and the naive B cells, and rarely also EBV infect T and NK cells. So EBV is very unique by its capacity to induce a very strong proliferation of infected B cells during the first steps of the infection. And these EBV-affected B cells strongly proliferate and are rapidly eliminated by CD8 cytotoxic T cells. NK cell and gamma T cells also play a role in the early phases of the immune response, particularly in children. And importantly, in healthy individuals, this EBV-specific T cell can represent up to 40% of circulating T cell in the blood, indicating that there is a massive and sustained expansion of EBV-specific T cell that is required to efficiently counteract EBV-infected proliferating B cells. However, some of these proliferating B cells will escape to the immune response, and there is a shutdown of the viral gene expression program in these cells, and these cells will become invisible to the immune system, and they acquire a B cell memory phenotype and return to the blood circulation in secondary lymphoid organs. and they will form the reservoir of EBV for all life. And EBV, the EBV infectious viral cycle can be reactivated in these B cells upon activation in the germinal center during differentiation into plasmocytes, causing new infection of naive B cells. And these reactivation, when they are not efficiently controlled, can ultimately result in a transformation and occurrence of B-cell lymphoma. So there are several pathologies that have been associated with EBV. So in most healthy individuals, the infection is asymptomatic. However, in some individuals which are immunocompetent, the primary infection of EBV trigger infectious mononucleosis, which is a self-limiting expansion of activated CD8 T cells, and EBV-affected B cell reflecting a strong immune response. And in contrast, in immunocompromised individuals, EBV infection can trigger several lymphoproliferative viral diseases, such as HLH, or virus-associated hemophagocytic syndrome, which is an inflammatory disorder associated with uncontrolled proliferation of activated CD8 T cells that produce large amounts of interferon gamma, and leading to secondary macrophage activation. The second type of disorder are the lymphoproliferative disorder, mainly the B-cell lymphoproliferative disorders that consist of non-malignant proliferation and B-cell lymphoma. As I told you at the beginning, EBV can infect T and NK cell, leading to T-cell lymphoproliferative disorder. Finally, EBV is also responsible associated with several non-lymphoid cancer and tumors, including epithelial tumors such as nasopharyngeal and gastric carcinoma. And finally, also EBV could cause EBV-associated smooth muscle tumors that are mainly developed in immunodeficient individuals. So this susceptibility to develop a severe EBV viral disease is often associated with immunodeficiency that can be acquired, like a patient with transplantation, or HIV-infected individuals, or inherited, like in patients with inborn error of immunities. And we can distinguish three groups of inborn error of immunity. Virtually all gene defects impairing T cell function can be associated with EBV susceptibility, or the susceptibility to develop severe viral EBV disease, considering the key role of T cell in the immunity to EBV. So in this group, the first group is a group that consists of defects that affect T cell, that impair globally T cell function. And in this group, there is 5 to 30% of patients that will develop EBV viral disease. So besides this group, there is two other categories of inborn error of immunity, which predispose to develop severe EBV viral disease, and this predisposition is particularly high. In one group, these patients are highly prone to develop B-cell lymphoproliferation and B-cell lymphoma. And these patients are carrying a gene defect impairing T-cell expansion, as you can see on the right of my slide. And in the other group of inborn error of immunity, patients are particularly susceptible to develop EBV-driven HLH and are less prone to develop lymphoma. And these inborn error immunity are caused by gene defect impairing the cytotoxic.
Can I just interrupt you for a minute? I don't think your slides are moving.
Oh, sorry. So, and in this group, in the last group, the third groups, this inborn error of immunity are associated with a high susceptibility to develop HLH and are less prone to lymphoma. And these speeds are caused by genetic defect impairing the cytotoxic function of T cells and NK cells. So based on this discovery of inborn error of immunity, they have revealed two critical steps of the immune response to EBV. One step is the expansion of EBV-specific T cell, which, when it's impaired, leads to accumulation of EBV-affected B cells and lymphoma. And the second step of this response is the effector phases, in which, when it's defective, you see cytotoxic defect, leads to proliferation of accumulation of CD8 T cells that are not capable to eliminate EBV-affected B cells. And in a normal response, there is expansion of EBV-specific T cells that allow to eliminate the EBV-affected B cell, and then there is contraction of this immune response and occurrence of memory T cells. This is the normal immune response. So over the last 10 years, my lab has been particularly involved in the identification and the characterization of gene defects that are associated with impaired T cell expansion of EBV-specific T cell. We and other labs have provided several lines of evidence that these genes form a functional cluster that is key for the expansion of EBV-specific T cells. Notably, this cluster provides signals and factors that are required for sustained expansion of activated T cells. So when one of these genes is impaired, is defective, the EBV-specific T cells are not able to proliferate and to expand, and this expansion is not sufficient to counteract EBV-affected B cells, leading to accumulation of EBV-affected B cells that could lead to lymphoma. And this cluster of genes, interestingly, some of these genes are expressed or upregulated at the surface of the B cell upon EBV infection, such as TNF-SF9 and CD70. And these genes are ligands for the co-stimulatory receptor of the TNF receptor family, CD27 and TNF-SF9, also known as 4-1BB, that are expressed on activated T cells. And this co-stimulatory molecule, directly or indirectly, activate downstream cascade when they are co-engaged with the T cell, the TCR, and activate downstream molecules such as the tyrosine kinase ITK, the exchange factor RAS, SHARP1. And these signals are important for the expression of CTPS1, which is a CTP synthetase involved in DNA replication in activated T cells, by producing CTP, which is a limiting nucleotide in activated T cells. We also show that the TNF-SF9, TNF-SF9R pathway is also important to control not only EBV-affected B cells, but also smooth muscle tumor, smooth muscle affected cells, and TNMP cells, EBV-affected TNMP cells. And more recently, we identified a defect in IL-27 array, revealing the role of the cytokine IL-27 in the terminal differentiation and expansion of EBV-specific T cells. Interestingly, this patient only presents severe infectious mononucleosis, as you can see here. So this patient all developed severe and persistent infection mononucleosis during primary infection, requiring hospitalization, but all, importantly, all have a favorable outcome without no clinical manifestation. For one, 15 years after the infectious mononucleosis. When we look at the immunological feature of this patient, they had decreased memory effector CD4 and CD8 T cell, decreased memory T cell, but normal IgG. So in this patient, we identified B allelic mutation in IL-20 in the gene IL-27 array, which codes for the which code for the alpha chain of the IL-27 receptor. So IL-27 receptor belong to the family of receptor cytokines that share the GP130 subunit. And IL-27 array is mostly expressed on lymphoid tissue. So in this patient, we show that this mutation leads to absence of expression of IL-27 array IL-27 array expression on the surface of the T cell, as you can see here. And when we look at the expansion of EBV-specific T cell in this patient, first we look at PBMC, the presence of EBV-specific T cell in the blood of this patient. We found that this patient have normal or increased level of increased number of EBV-specific T cell, as you can see here in these two patients. But when we co-culture these PBMC with EBV, autologous EBV transformed B cell line, these EBV-specific T cell failed to expand, as you can see here. And this impaired expansion was associated with acquisition of activation marker, abnormal acquisition of activation marker, and marker of exhaustion. And this cell failed to kill autologous B cell line, as you can see on the right of my slide. So what we know about IL-27, we know that this receptor when bound to IL-27 activates the JAK-STAT pathway, particularly STAT1 and STAT3, and it depends on JAK1 and JAK2 kinases. And early studies on mice have shown that IL-27 favor TH1 differentiation and promote T cell proliferation of naive CD4 and CD8 T cell. And more recently, expansion of CD8 T cell have been found to be IL-27 dependent in persistent viral infection in mice. So first we look at signaling in T cell of one patient, signaling downstream IL-27 array. As you can see, there is in the patient that is IL-27 array deficient, there is no phosphorylation of STAT1, either STAT3, upon IL-27 stimulation. And we also analyze the capacity of IL-27 to trigger T cell proliferation, as it was reported in previous studies that IL-27 was important for the expansion of activated T cell. In our ones, we found that IL-27 alone has no effect on proliferation, as you can see here, but it has a strong synergic effect when you stimulate T cells with low doses of anti-CD3 antibodies, as you can see here. This is the gray histogram. And as expected in patients, you felt this synergic effect in red. Interestingly, in co-culture experiment in which we look at the expansion of EBV-specific T cells in the presence of autologous EBV transformed B cell line, we did not add exogenous IL-27, suggesting that the IL-27 in this assay was provided by the EBV transformed B cell line. So importantly, this could tell us why IL-27 pathway is so important and specifically involved in the immunity to EBV infection. EBV being the trigger of IL-27 production by B cells when they are infected by EBV. To test this hypothesis, we took control PBMC and we infect this control PBMC with GFP EBV strain. As you can see here, we are able to infect B cells, which are CD19 positive, and we look at IL-20 production by intracellular staining. As you can see, the B cells that have been affected by EBV express IL-27, while the B cells that have not been affected by EBV are negative for IL-27. And further supporting this observation that EBV-infected B cells, that EBV trigger IL-27 production by B cell, we show that by ELISA that IL-27 progressively and significantly accumulates in the supernatant of PBMC affected by EBV on all EBV transformed B cells that we tested produce a large amount of IL-27. And this production is really important for the expansion of EBV-specific T cell in vitro, because if you block the IL-27 produced by the EBV transformed B cell line using anti-IL-27 or soluble plus soluble IL-27 array recombinant protein, you see that you block the proliferation of EBV-specific T cell, and this T cell acquire exhaustion and activation marker like we observe in cell of the patient. We also observe in course of these experiments that EBV transformed B cell lines obtained from patients have decreased growth compared to EBV transformed B cell line. As you can see here, this cell in red, this is the cell from the patient, they grow slowly and they proliferate slowly compared to control. Interestingly, control LCL express significant amount of IL-27 at their surface, and when you stimulate the control LCL with IL-27, you induce a nice phosphorylation of anti-phosphostatin and anti-phosphotat3, meaning that this pathway in normal B transformed B cell line is active and functional. So we also tested EBV transformed B cell line derived from patients with a STAT1 deficiency, and as can be expected, this cell line also behave as an IL-27 array deficient B cell line. They fail to grow, they fail to expand. So therefore, this result suggested that the IL-27 array pathway is functional in EBV transformed B cells and is involved in the growth of the cell line, possibly through an autocrine loop as LCL produce IL-27, as I showed you before. And to test and formally prove the existence of this autocrine loop, we did two things. First, we co-culture control LCL in presence of blocking neutralizing anti-IL-27 antibodies, and you can show here that these antibodies, when they add to the culture, they block the proliferation of control LCL, which is not the case if you use an anti-IL-2 antibodies. And the second thing that we did, we inactivate the IL-27 gene by CRISPR-Cas9, and cells were maintained with IL-27. Here you see the cells that have been CRISPRized for IL-27. And when we remove IL-27, the cell rapidly die in culture. So this result indicates that IL-27 is required for the growth and the maintenance of EBV transformed B cell line via an autocrine loop. And importantly, this could explain why patients have not developed further B cell lymphoproliferation disease as EBV positive lymphoma or B cell lymphoma following their infectious pneumococcal. So finally, I would like to finish with an interesting observation that we had in this study. As you know, there is now accumulating evidence that the existence of the role of anti-cytokine autoantibodies in several infectious diseases, like phenocopy, inborn error of immunity that predispose to these infectious diseases, such as autoantibodies raised against L17 or type 1 interferon. So therefore, we thought that maybe neutralizing antibodies could exist, particularly in individuals who develop sporadic infectious pneumococcal disease, while these individuals are considered to be healthy and well. So we collected a serum sample for 15 individuals with sporadic infectious pneumococcal disease requiring hospitalization and/or immunosuppressive treatment in most of these cases. We also analyzed patients with chronic EBV infection, in which EBV-infected T and NK cells. And to detect these antibodies, we developed an ELISA-like assay in collaboration with NPL at Imaging Institute. She also provided us with serum sample of patients with APSED syndrome and STAT1 gain of function patients that are known to contain, these serum are known to contain high concentration of autoantibodies raised against different cytokines. Using this assay, we are able to detect significant level of autoantibodies raised against IL-27 in sample of patients with infectious pneumococcal, chronic EBV infection, and patients with STAT1 gain of function, while these levels were low or undetectable in healthy control or patients with APSED. As you can see, these antibodies, we verify that these antibodies containing this serum were able to recognize recombinant IL-27 by Western blotting. This is the case, as you can see here. These autoantibodies are able to detect recombinant IL-27. We use as positive control a monoclonal antibody with, again, IL-27. We also measured the level of IL-27 and the complexes of autoantibodies with IL-27 in all samples. And we found that autoantibodies raised again IL-27, as well as the complexes, the immune complexes, are positively correlated with the level of IL-27. And finally, more importantly, we show that these autoantibodies that we detect in patients with infectious immunodeficiencies have the capacity to neutralize and inhibit the synergic effect of IL-27 proliferation of activated T cell, as it's shown here in this proliferation profile, in which we add to the proliferation of CD4 T cells these autoantibodies, this serum. As you can see, the serum from a patient with infectious immunodeficiencies, chronic EBV infection with STAT1, clearly block the proliferation compared to the control serum. And if you accumulate this experiment of proliferation, you could calculate expansion index of this CD4 T cell. And as you can see, all serum samples that we tested, which each point is a serum sample, all serum samples containing IL-27 autoantibodies that we tested were able to block the proliferation of CD4 T cell compared to control serum. So IL-27 deficiency is a novel inborn error immunity associated with selective susceptibility to EBV infection, and particularly to develop severe primary infection. And this patient seems not to develop other symptoms than this severe primary infection. And we show that this pathway is important to promote the expansion and the direct differentiation of effector EBV-specific T cell. And IL-27 is produced by EBV-infected B cell upon infection, and so this production may drive the expansion of EBV-specific T cell in vivo in the context of the primary infection. And we also show that autocrine production of IL-27 is necessary for the maintenance and growth of EBV transformed cell line in vitro, and this possibly might explain the favorable outcome of the EBV viral disease in IL-27 array deficiency, who did not experience relapse of EBV-driven proliferative episode of EBV-positive lymphoma until now. And finally, we show that individuals having experience of severe infectious mononucleosis or presenting chronic EBV infection have detectable anti-IL-27 autoantibodies with a neutralizing activity. And these autoantibodies could represent a phenocopy of the IL-27 deficiency. So I would like to acknowledge all the people on my labs that have been involved in this project, particularly Emmanuel Martin and Sarah Winter, as well Benedict Devan, and for the collaboration and QL. So I stop there.
So thank you very much, Sylvain, for that absolutely fascinating talk. Can I remind people again that if they have questions, please put them in the Q&A, and we can then address them at the end of the webinar. So meanwhile, we have our third and final presentation from Benjamin Fournier, who is works at Necker in pediatric immunology, hematology, and rheumatology, and also works with Sylvain Latour, looking at the interactions between EBV and the immune system, particularly in EBV immunodeficiencies and TNK lymphoproliferative disorders. So Benjamin will present EBV-associated inborn errors of immunity and associated disorders clinical aspects and treatment. Thank you very much.
So thank you very much for the invitation. So can you hear me and can you see my screen?
Yes, we can
Okay,
hear you and we
nice.
can see your screen.
Yeah, perfect. So I will have a more broad view of EBV-related disorders in inborn error of immunity related to clinical aspects and treatment. There will be some overlap with Sylvain talks, but I will try to be quicker on these parts. So I will, as you previous speakers have mentioned, EBV infect during primary infection B-cell lymphocytes, but EBV-driven disease are mainly related to EBV-infected B cells, such as Hodgkin lymphoma or Burkitt lymphoma, or during primary infection during infectious mononucleosis. But there are also diseases where EBV is infected T or NK cells, and I will try to talk about both of these subsets of diseases. So here we can see on the left side, I will talk about EBV-infected B cells, and here the T cells are also involved in the clinical phenotype of the patient. On the right side, you will see EBV-infected T cells, and here it's mostly only T cells or NK cells that are involved. So when EBV infect B cells, you may be in the context of primary or secondary immune deficiency. So primary, it's related to an inborn error of immunity with a genetic defect, and secondary immune deficiency, for instance, during solid organ transplantation, when you have treatments that impede T-cell response. And in this disease where EBV infect B cells, this is secondary to a T-cell immune defect, which are unable to control EBV-infected B cells, as Sylvain has previously mentioned. And we can distinguish two subsets of disease. Disease where the clinical phenotype is due to T-cell immunopathology, such as fulminant infectious mononucleosis or HLH, where we have a T-cell infiltration that will cause the symptoms. And on the other side, you have phenotypes which are more related to, let's say, hematological disorder, which are caused by EBV-infected B cells, which are more related to B-cell lymphoproliferation. And when it is associated to inborn error of immunity, you may also find features of autoimmunity, autoinflammation, or allergy or susceptibility to infections. So on the right side, you have EBV that in these instances will infect T or NK cells, and these are called EBV-positive T NK cell lymphoproliferation. And here it's mostly hematological disorder rather than primary immune deficiency. And the clinical phenotypes of the patient will be HLH, more or less T-cell lymphoproliferative disease, and in some patients who have T or NK cells CABV, chronic active EBV infection, these patients may present with disease that looks like dysimmune or inflammatory disease. So in both subsets, you have in most cases, elevated blood PCR, but there are different disease mechanisms, and it means that the treatment will be different. So we'll start with the disease where EBV infect B cells, and I will be quick. I will be quick on that because Sylvain has already showed you this kind of slides. Here is to show you the anti-EBV immunity where EBV infect B cells with B cells in red and the T cells in purple. So then the T cells will enter in close communication and form a synapse with the infected B cells through co-stimulation molecules and TCR. And then you will have this strong T-cell proliferation, and then with the expansion phase, which will lead to the effector phase, where the T-cell will use perforin and granzyme to kill the infected EBV B cells. And if we look, as Sylvain has mentioned, to a defect in both of these phases, if we start with a defect in the T-cell expansion phase, it will lead to a phenotype of EBV-positive B-cell lymphoproliferation, so lymph nodes, B-cell infiltration of organs, and this is due to a T-cell defect in proliferation, which can be lead to genetic disease. You have here all the genes that are involved in combined immune deficiency. You have here the genes that are involved in B T cell co-stimulation defect, including Pure-Tilo syndrome, but it may also be due to extrinsic T cell defects, such as post-transplantation lymphoproliferative disease, where the T cell activation is due to cyclosporin A or corticosteroids to treat transplant rejection. And because T cells are unable to proliferate, the B cells which are infected by EBV will proliferate because of the virus, and this will lead to what we could call B cell chronic active EBV infection. And then over time, these B cells may accumulate somatic events, and this may lead to overt lymphoma such as Hodgkin or Burkitt lymphoma. Here is an example of a young patient with a CD27 deficiency, which is a co-stimulation molecules. He was 6 years old with hypogammaglobulinemia because the B cells require an interaction with the T cell to be fully functional. The patient has a very elevated chronic EBV PCR nasal load, and he had clinical proliferation with lymph nodes, pulmonary involvement, masses. This proliferation was polyclonal B cells and of course EBV positive, and he went well after allogenic hematopoietic stem cell transplantation. Here another example with ITK deficiency, which is more related to combined immune deficiency. You can see that the EBV load in the blood is extremely elevated. This is strongly indicative of primary immune deficiency. It starts at a very young age, and there is a lymphoproliferation in the lymph nodes, bone marrow, in the blood, but also what is more typical of ITK deficiency, you can see here acute respiratory distress syndrome because there are B cells in the lungs. And what is interesting is that when you remove infected B cell using rituximab, there is a strong effect, and the patient became much healthier, and the EBV load remained negative. But here the problem is that you did not correct the genetic defect, and the T cell defect is still there. And if you do nothing, there will be probably a new lymphoma, as in this patient, and she developed a new Hodgkin lymphoma. She was treated, and then you have to correct the genetic defect and go to allo-HSCT. So the second group of disease related to EBV-infected B cells are related to HLH and severe EBV-related infectious mononucleosis. And this this is related to a defect of the second phase, which is related to T cell cytotoxicity. And here, this is due to genetic defects for the cytotoxic machinery and granules defect. And here you have all the gene of familial HLH. You have also the gene of Pure-Tilo syndrome here, XLP1. And what happens here is that T cells are unable to kill EBV-infected B cells, but they are still able to proliferate, and they will proliferate. They will secrete cytokines, but they will fail to kill EBV-infected B cells, but they will still try to kill these infected B cells, and you will end up with a positive feedback loop, and you will end up with very severe fulminant HLH. And maybe the last disease here is XIAP deficiency. Here, probably the defect is also more related to myeloid dysfunction, as also these patients have autoinflammatory disease. So here we have the example of a patient with XLP1. So it's a boy because it's X-linked. He was very well before encountering EBV, and at 17 years old, during primary EBV infection, he developed severe peripheral and central HLH. You can see that the EBV load in the blood was quite elevated, and you can see over time that he had hepatitis, fibrinogen was not so low, increased ferritin, and you can see that he had also HLH in the CNS, as you can see here in the brain MRI. So it was treated by dampening T cell activation with corticosteroids and cyclosporine. You have to also remove the infected B cells, and here it was not enough to control the HLH. So we get rid of all T cell using alemtuzumab and dampening also T cell activation using JAK inhibitors. And maybe you have seen in my slide related to B-cell lymphoproliferation that the gene of the Purtilo syndrome was also present in these B-cell lymphoproliferative diseases. And the same patient, after having removing T cells, in fact, he developed a B-cell in EBV-positive B-cell lymphoproliferative disease with a monomorphic lymphoproliferative disorder with, you can see, lung involvement and also liver involvement. And then he had a favorable outcome after allo-HSCT. So in this disease where EBV infect B cells, what could be the treatment? So in EBV positive B cells associated lymphoproliferation, when you have an enlarged lymph node, hepatosplenomegaly, histology is often atypical. Sometimes you have combined immune deficiency. What you have to do is to remove infected B cells using, for instance, rituximab, and then you have to go also to allogenic transplantation, bone marrow transplantation. Regarding EBV B cell associated HLH related to cytotoxic defect, you have also to remove a trigger. So you remove EBV using B cell depletion such as rituximab, but you have also to decrease T cell activation, and for that you can use corticosteroids, ruxolitinib, and you can also completely remove T cell using alemtuzumab or using etoposide, and then you also have to move to HSCT. So in the beginning, I told you that there is subset where EBV infect B cells and these other diseases where EBV infect T cells, and it is important to make the difference between both diseases. So how can you determine that EBV is in T cell or B or NK cells? So what you can do is to sort PBMC, you sort T, NK, and B cells, and in each fraction you perform EBV PCR. So it's quite cost effective. Maybe there is a risk of false positivity if you have some T cells that go in the B cell compartment of some infected B cells that go to the T cell, you may have falsely positive fraction. What we tend to use here nowadays is Eber-Flowfish, which is also based on blood samples, so it's easy to get. There is a fewer risk of false positivity, and it uses flow cytometry, and it combines an Eber probe that will target this EBV RNA that is present in all infected cells, combined to extracellular markers to see if you are in B, T, or NK cells. The drawbacks is it's quite expensive, and it's today's experiment, but it remains a robust way to find where EBV is located. And finally, importantly, histology is very important also, and you have to do co-staining on the same slice to have an EBV marker on T or NK cell or B cell marker. But for that, it requires a tissue to be biopsied. And here is an example of what we can have by Eber-Flowfish, by flow cytometry. So you have the Eber staining here. The x-axis, when you have on the right side, you are infected by EBV. Here is a B cell marker, and you can see here during primary infection, during Purtilo syndrome and macturan deficiency, you have EBV that is only located in B cell, and there are a lot of cells that are infected during XLP1 primary infection. And you have also here other example of combined immune deficiency, and as you can see in ZAP70 and CD70 deficiency, you have also EBV almost only in B cells, and this is the same kind of picture that you can have in proliferation after a solid organ transplantation. And you can see here that there is quite a little percentage of infected B cells, but this is enough to have a quite elevated EBV PCR in the blood, in whole blood, I mean. So having said that, I will go through the second subset of diseases where EBV infect T and or NK cells. So these are classified in the World Health Organization classification of lymphoid neoplasms. And what is the core feature, of course, is that EBV infect T and or NK cells. We have a first subset, which is called T NK cell type CABV, so for chronic active EBV infection that occurs long after primary infection. There are two subsets, a skin subset, which encompasses vacciniformis and severe mosquito bite allergy, and a systemic form with heterogeneous lymphoproliferative disease, which can mimic these immune or inflammatory phenotypes. And as we'll see, what is very difficult in these patients is that they have an unpredictable course, and they may be very well for months or years, and very suddenly they can develop fulminant T-cell lymphoma or fulminant HLH, and it's almost impossible to cure this patient at this time. And another subset of EBV positive NK cell lymphoproliferation, this time it occurs at primary infection, and EBV infected T cells, and it is called systemic EBV positive T cell lymphoma of childhood, and the patient will present with a very severe HLH. So in all of these diseases, you can find EBV infected T or NK cells in tissues, but there is always the same subset that is present in the blood. So it's a good way to have blood to make the diagnosis. So and you have to get the proof that it is T NK cells using the methods that I've previously mentioned. So here is an example of systemic EBV T cell lymphoma of childhood. As I mentioned, is during primary infection with fulminant HLH, multi-organ failure, generally with poor outcome. EBV is only in CD8 T cells, as you can see in both patients, one patient and here a second patient. There is almost nothing, if anything, in B cells, and all is located in CD3 and more particularly CD8 T cells. And the differential diagnosis with HLH at primary infection, severe HLH at primary infection, is Peutilo syndrome and familial HLH. Then there is this difficult to, let's say, understand and to treat a systemic CABV of T NK cell type. It occurs long after primary infection. So far, it's most common in Asia, but in fact, probably it's underdiagnosed in Western countries. So Japanese teams have established diagnostic criteria. For this patient, EBV load is always very elevated in the blood. You have to prove that there is an infection of T and NK cells in tissue or in blood. And this patient have systemic inflammatory symptoms for more than 3 months, but it's mostly for months or year with a spontaneous relapsing remitting course. And as I mentioned, there is no immune deficiency which are associated to this disease. And here I will show you some example of patient which can mimic inflammatory disorders. Here is a patient that started the disease at 21 years old. And with polyarteritis nodosa, you can see here the aneurysm in the mesenteric arteries and the digestive arteries. You can also see here it's the vertebral arteries with aneurysm. He also had intermittent fever. He had a mild hepatosplenomegaly. And he had a severe outcome because he has brain hemorrhages. And he had increased EBV PCR in the blood. And in fact, all the EBV virus was located in gamma delta T cells. And it was also the case in the liver biopsy. Here you have another patient with diagnosis at 11 years old. It was diagnosed with EBV only at 3 years later. And he has a phenotype of inflammatory bowel disease, but as you can see here in the MRI, but you can see also there are a lot of lymph nodes here, and he had severe diarrhea, increased inflammatory markers, infected NK cells, both in the tissue and in the blood, and unfortunately he died after HSCT. And here it's also to show you the very difficult to predict course of this patient. Here you have a patient with recurrent fever since he was 7 and 10 years old. He was thought to have autoinflammatory disease, but nothing was found on the genetic tests. At 15 years old, because of mild HLH, EBV was tested, and in fact, it was very elevated in the blood. In the liver biopsy, it was shown that it was in NK cells. He had only a short course of corticosteroids, and then he was extremely well without any flares for years, but he had still a very elevated EBV in the blood. And at 20 years old, he died very suddenly of very fulminant HLH, despite all the treatment that could be given to him. So very difficult to predict who should be treated and when. So regarding the pathophysiology of this disease, we previously discussed the effect in the EBV genome, but probably it's more related to hematological disorder. Here you can see that there are somatic mutations that are related to T and K cell lymphoma that are present in this cohort of Japanese patient. Here you have DDX3X, KMT2D or TET2 mutation, which are also present in this Chinese cohort. And I can also tell you that we have a cohort of similar patients from Europe and Africa, and they are both the same kind of somatic mutation in their infected cells. So what are the treatments for this patient? So these patients have a severe prognosis because, as you can see, they may have severe vasculitis, severe IBD, and there is a high risk of transformation, but sometimes after years of evolution. So how can you treat them? You may use maintenance therapy to try to dampen T-cell activation, but it will never be curative. You will never get rid of EBV using JAK inhibitor, cyclosporine A. Maybe anti-PD-1 could be tested more often. And the only curative treatment is allogenic stem cell transplantation, but with a very high morbidity. And what is difficult is to propose to the patient that is very well a very difficult treatment. So I will try to be faster on the last slide. The Japanese teams have a strategy where when they do the diagnosis of CIBV, they start to dampen T-cell activation using cyclosporine A and etoposide and corticosteroids. We use more ruxolitinib rather than cyclosporine A. Then they go through chemotherapy, and all the patients have allo-HSCT. So maybe in the future, there will be other therapeutic that will be interesting, such as anti-EBV CAR T cells. And here it's a result of allo-HSCT in Asian patients, and here it's United States. You can see that the overall survival is not so good, but it's better when you have HSCT. Here, the overall survival is not so good, but probably because the patient was diagnosed long after diagnosis. And here, finally, you have my last slide to show you the last results for the Japanese team that use a registry. You can see that overall, the percentage of survival is better, but is not so good after HSCT. Pediatric patients seems to have a better outcome. And what is very important is that you should try to have an inactive disease at diagnosis, because the prognosis is much better when the disease is controlled at HSCT. So I will finish there, and thanks all the clinicians in the department and also Sylvain Latour in the lab. So thank you for your attention.
So thank you very much for that. Again, fascinating talk. Could all the panelists turn on their cameras, please, and their microphones? And then we have some questions. Can I remind everybody, if you've got questions, please put them in the Q&A and not in the chat. We do have some in the chat, but which I will try and read out as well. So I think that most of the questions are around management of EBV-associated malignancies and conditions. So I guess that most of them are going to be directed at Benjamin, because neither of the, I don't know, Sylvain, is that correct? You're not somebody who's managing patients with inborn areas of immunity.
No, I'm not managing patient. I'm doing basic science, so Benjamin probably could.
So I'm not I mean, I'm not directly managing, but I can probably add something if Benjamin has something to say. Okay, so we have the first question is, given the increased risk of EBV-associated malignancies in inborn errors of immunity, are there any biomarkers or clinical features you'd find for particularly useful for early detection? And actually, that might be something that Sylvain might want to ask, might want to answer as well. But let me start with Benjamin.
Yeah, thank you. This is very difficult. No, so far I think we don't have good biomarkers to know when a patient with a known inborn error of immunity is having a malignant transformation. First of all, in inborn error of immunity, you may have very well-characterized lymphoma, such as Hodgkin lymphoma or Burkitt lymphoma, such as with Peutilo syndrome. But in most of the cases, the histology is very atypical, and it's difficult to classify the lymphoproliferation. And thus, it's also difficult to say that it's a true lymphoma, and you have to go through chemotherapy. So this is the first thing to say. And in terms of biomarker, for instance, in ataxia telangiectasia or PI3 kinase deficiency, these patients are at risk of EBV-driven B-cell lymphoma, and they often have increased EBV load in the blood. But you can't rely on EBV load in the blood to tell if this patient is doing a clinically relevant lymphoproliferation that would deserve a treatment. So even with that, it's difficult to use EBV load in the blood. So in the end, probably you have to stay on the clinical phenotype of the patient, and if he has symptoms, and perform a CT scan, try to have a biopsy, and to characterize the more deeply than you can the lymphoproliferative disease. So the short answer is, I think so far we don't have good biomarker to tell that your patient with inborn error of immunity is having a malignant proliferation.
Okay, I'm sorry, go on, Sylvain.
Maybe we could say that other clinical symptoms on EBV susceptibility could be associated with this inborn error of immunity, such as autoimmunity or other type of symptoms that could say that this patient has an inborn error of immunity.
Yeah,
okay.
It depends on the question. If the question is in front of a patient with a lymphoma, when should you think of inborn error of immunity, or in front of a patient with a known inborn error of immunity, when should you think of lymphoproliferative disease, I think. But I agree with you, in patient with Hodgkin lymphoma, there are several hits that could make you think of primary immune deficiency.
So can I broaden that out? When would you, what do you think, I mean, and Sylvain, this is for you, from the work that you do, which are the areas that you think will be most translatable into biomarkers? I'm particularly struck by your IL-27 story in infectious mononucleosis and chronic active EBV. Do you think that is a potential biomarker that we should be looking for in patients with who present with those conditions?
We have clear answer today. Just a correlation. So we identify these autoantibodies in patients with severe infectious mononucleosis, but we don't know really what is the role of these autoantibodies and in the physiopathology of the severe infectious mononucleosis in this patient. We really don't know if it's playing a role, but for sure in vitro, these antibodies are neutralizing and they block the IL-27 capacity to expand EBV-specific T cell. But in vivo, we really don't know.
And you have no idea of the prevalence in patients with infectious mononucleosis generally, or in presenting with chronic active EBV?
All patients that we tested with severe infectious mononucleosis, I say severe, I mean that they have been hospitalized for a few days and they have received an immunosuppressive treatment. In all these patients, we detect autoantibodies.
Okay.
And in patients with chronic EBV infection, I think it was the same also. We detect all these autoantibodies.
Thank you. Anything to add to that, Benjamin, since I know you were involved in that work?
But I was thinking in terms of biomarkers, maybe also the genetic alteration of the EBV genome, as you mentioned, maybe during primary immune deficiency, if maybe could be interesting to study if, for instance, in patients that has a stable increased EBV load in the blood with no over lymphoproliferation, to see if there is defect in EBV genome and to compare it if in the end it developed lymphoma, if at the same time, both in the blood and also in the tumoral tissue, this time you detect genetic defect in the EBV genome. Maybe it could be an interesting biomarker.
Cristina, have you? Sorry, carry on.
Sorry. No.
Finish what you had to say.
So, and what may be also interesting, maybe in the future, is liquid biopsy, probably to look at somatic mutation of the human genome, or maybe of EBV genome that are released from the tumor in the blood. Because I guess I don't know, but probably the B cells that are circulating in the blood are maybe not the same that are driven the tissue lymphoproliferation.
Cristina, do you want to comment on that, on what we find when we look at the genome in B cell lymphoproliferative disorders and PTLD compared to the T cell and NK cell lymphoma?
Yeah, so I mean, I think one piece of that perhaps I should have underlined more when I presented is that when we actually look at the blood of the PTLD, we don't actually see deletions, but we do see deletions, I think, in all cases of T and NK cell lymphoma. And I think what is actually quite interesting, and what we found actually related more with the outcome, is the persistence over time. I think what would be quite useful is to have longitudinal data where you can see a little bit over time what's happening, rather than have the one sample, because that's for what we could see, we couldn't really relate the presence or not with specific outcome. And the other interesting bits was that I mean, we need to look at the blood or the biopsy rather than the saliva. The saliva is not actually particularly good for this sort of biomarker. So that's the other bit that is quite interesting.
Yeah, and in fact, we find that the B cell, that the EBV is actually clonal in B cell lymphomas. It's actually clonal in the blood, whereas in TK lymphomas, it's heterogeneous and with a subset that have deletions, so that that subset can expand as the patient becomes unwell, has been our experience. Right, going back to the chat, there are some specific questions. Treatment for EBV positive T cell lymphomas, does rituximab work in these patients? Should we think of EBV specific T cell therapy?
So I guess it's for me. No, rituximab, it does not work, and it's a good indication that EBV is in T cells. If you use rituximab in this patient and you still have a high EBV load in the blood, despite having depleting all B cells, probably that EBV is not in B cells, but in T or NK cells. So no, rituximab won't work for T and NK cell related lymphoproliferation. Then depleting T cells, it depends on which subset of, I think, EBV positive T and NK cell lymphoproliferation you are. In case of these EBV in T cell during primary infection, it seems that there may be different subsets with overt, very difficult to treat lymphoma. And in this case, depleting only T cells using alemtuzumab won't be enough. And there is also what seems to be called in the literature EBV-driven HLH. And here it seems that this is less severe disease, and maybe with only etoposide, you may control the disease and it may end. And what is very difficult is to treat chronic active EBV infection. And this patient, using just alemtuzumab, it doesn't work. The EBV infected T cells will come back because it also come back even after allo-HSCT with myeloablative conditioning regimen and T cell depletion therapy to prevent graft rejection. So this is, I think this is a real challenge, is to know how to treat this patient. And so far, the only curative treatment is allo-HSCT, and deeply depleting T cells is not possible because you can't live without T cells. And so probably the message is to find a good target that would deplete only infected T cells. But maybe anti-PD-1 could be interesting. But it's probably a combination of both chemotherapy and anti-PD-1 that could work, as is seen in extranodal TNK cell lymphoma, I would say.
Okay, an interesting question about pathogenesis, interplay between EBV and patient somatic events, particularly in the TNK subtype. How often do these occur? And is there some mechanistic insight into how these may cause the uncontrolled proliferation? And I'm guessing also that the interplay between the deletions that we see and the somatic mutations may also be of interest. Sylvain, do you have any views on that?
They probably interplay between the EBV genome and some of these somatic mutations. No, I have no clear answer to that. But it's clear that the somatic mutations are really important in this disorder. Inborn error of immunity are not really associated with this type of disorder, rarely, but this disorder are mainly driven by somatic mutation, as Benjamin said.
Yes, so we don't see the deletions in the EBV genome in quite the same way in the inborn errors of immunity, and it's interesting that the somatic mutations are also not present. I mean, obviously you have the germline mutation too. Benjamin, any suggestions as to what might be happening in that situation?
It's difficult. I think probably there is an interplay. I think what has been published is that all patients do not, for instance, in TNK cell lymphoproliferation, these are not all the patients that carries a somatic mutation. And these are not also all the patients that carries EBV genome deletion. And probably, I guess it's an accumulation of events that will lead to the clinical phenotype. We think it's based on the germline mutation, which favor maybe T cell infection and T cell proliferation, and maybe there is an accumulation of events that will lead to the clinical phenotypes that involve both EBV genome and somatic mutation. And what I think is not perfectly known is when does the EBV infection occurs? Does it occur in a cell that has already a somatic mutation, and then it helps to maintain the T cells proliferation and so on? Or does it occur in a cell that has no somatic mutation, but it helps to maintain the T cells and then it accumulates somatic mutation? I think so far we don't know which is the timeline of events.
One interesting thing that we have found is that the virus that's present in saliva is intact, and the same virus in the T cells can have these deletions. So it's clearly something that's happening, clearly specifically in relation to the infection of certain cells. And yes, your explanation would be good.
What we know about these large deletion, they provide a selective advantage of the cells? Or?
You're asking a question.
Yeah, I'm asking what we know about this large deletion. What is the role of this large deletion?
We know that they are not compatible with replication, and we think that they do provide a selective advantage in the sense that they confer immune escape to that viral infected cell. They're not actually they allow that cell to escape the surveillance of immune surveillance by down regulating immune active EBV genes. And it's possible that the somatic mutations are actually possibly doing the same to the cell. So who knows? I think we're running out of time. So I think that there are some other interesting questions, but I don't think we're going to have any more time to answer them. So it just remains for me to say thank you to everybody for attending this webinar. Thank you to our speakers, Cristina Venturini, Sylvain Latour, and Benjamin Fournier, for excellent talks, which really stimulated a lot of interest. I've been asked to mention 3 events that are happening in the ESID community. The first is this symposium, and you can see the details of it in Vienna in November, and you should have submitted the abstracts by now, but I don't know whether there's a late abstract submission, but anyway, that's happening. The next event is the new peer-reviewed Journal of Human Immunity, which is being put out by a consortium of which ESID is a founding member, and it's being edited by Jean-Laurent under the editorial guidance of Jean-Laurent Casanova, and assisted by Megan Cooper. So I guess the encouragement is to try and publish your ESID related material in this new journal. And then finally, to mention that there is an upcoming junior clinical cases webinar, which will take place on the 21st of October with a number of speakers, and it's looking at unusual pathogens in inborn errors of immunity. So I think you're being encouraged to sign up for that, especially juniors who wish to have exposure to these interesting cases. So with that, I think I can only say, it remains me to say thank you very much, and have a good rest of your evening until next time. Thank you.
Thank you.